Bismillah ar-Rahman ar-Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. Today in a series of video lecture we are going to talk about MHC molecule in detail. Okay, so let me write MHC molecules. Now this is the first video lecture of MHC molecule, and in this first video lecture we are going to talk about introduction, the overview or the big picture of the MHC molecule. Once we get the idea about the MHC molecule, then the rest of the video which are regarded to MHC molecule will be very easy for us. Okay, so first of all, let's try to understand about this word, that what are actually MHC. Now this MHC stand for major histocompatibility complex. Let me write major histocompatibility complex. Now what does it mean? So this major means specific, histo means tissue, compatibility means against or having the ability along with, or we can say having ability. And this complex means a process, a complicated process, we can say like that. Although it is very complicated topic, but I will explain this topic in easy way. Now, what are MHC molecule? These are just the meaning of MHC molecule that I write here. Okay, now what are actually the MHC molecule? So first of all, let me explain that how our body can distinguish between the self and non-self antigen. Okay, now there are special kinds of receptors, special kinds of proteins, special kinds of molecules which are present on the cell surface. Each molecule are specially responsible to degrade, to kill, to recognize the non-self antigen, which are especially which are taken from outside of the environment by our body. And there are also self antigen are also which are recognized by the surface molecule which are present on the membrane. So each surface molecule are called as a MHC molecule. Okay, so if I draw the cell, means all the eukaryotic cells in our cell, all the eukaryotic cells have MHC molecules. So if I draw the cell here, let's suppose, so let's say this is a normal cell of our human body. Now on the surface, let's say these are the surface of the eukaryotic cell. So on the surface of the eukaryotic cell, there are special kinds of receptor or special kinds of globulin or immunoglobulin or proteins, special kinds of proteins which we call that MHC molecule. Okay, now how this MHC molecule are synthesized? Now what are the function of each MHC molecule? Let me first of all explain that the function of each MHC molecule. MHC molecule are specific to the tissue. Let's suppose if there is a special kind of tissue which is present in my body, or there are special kind of organ which is present in my body, let's suppose kidney, liver, and we can say that eye, these kinds of organ, let's suppose, is present, but it is not killed by our own eukaryotic cell or our own amino cell. It is usually these amino cell, let's suppose this is the amino cell. Okay, this amino cell can only kill or can only recognize, or these amino cells are against the foreign body. If the foreign organ or the foreign tissue are transplanted to us, so then our amino cells are activated and they are against to each of the molecule or to each of the organ. And how they recognize? They recognize each of the organ by this receptor or by this molecule which we call that MHC molecule. So now how they can recognize the means the self antigen. So means non-self antigen, how they recognize. Let's suppose there is an amino cells, let's say any kind of amino cells, they recognize the antigen. Or let's suppose we have a phagocytes in this case. Okay, they could be a macrophages, dendritic cell, or any kind of the phagocytes we have. So they get let's suppose this green one represents the bacteria. The bacteria are engulfed by this phagocytic cell. Now once they engulf the phagocytic cells, so the phagocytic cells are then converted into small pieces, means they are digested. Okay, once it is digested, so then it is transferred to the proteosomes, which is a kind of means a machine which are specially responsible to break down each of the molecules. Now once proteosomes break down each of the molecule, which is already converted into small fragment, after that these molecules are shifted to the endoplasmic reticulum, and after the endoplasmic reticulum there are the MHC molecule are synthesized. Now once the MHC molecule are synthesized, let's suppose, so then each MHC molecule are shifted to the cell surface. Now once it is shifted to the cell surface, so after that it presents each of the bacterial portion. Okay, so it presents each of the bacterial portion. Let's suppose any kind of portion, lipid portion, flagella portion, or any kind of portion they are representing. And after some time there are another cells which we call that helper T cells are come to closer to each of the cell. Let's suppose this is the helper T cell. Helper T cell. They recognize what? They recognize each of the antigen. Now once they recognize the antigen, so these helper T cells are divided into more helper T cell. So let me draw. They divided into more helper T cell. This more helper T cell can produce cytokines. Due to each cytokines, they will call upon the B cells. So we get the B cells. After that, B cell is converted into plasma cell. Now let's suppose this is the plasma cell. Now after that, this plasma cell produces a special kinds of antibody which is specific to these kinds of antigen. Okay, so if there is other bacteria, same bacteria, if it is present in the surrounding of our body, so these antigen, so this antibody will against to each of the bacteria, and finally the process are under the control. Okay, so that's how each of the process are happening inside our body. But sometime what actually happen, these kinds of helper T cell will call upon the cytotoxic T cell by producing another cytokines. So they produce some other cytokines due to which they can call upon the cytotoxic T cell. Now what are the function of cytotoxic T cell? Cytotoxic T cell have the function to recognize this, and this cell is infected. Why? Because they can digest the bacteria, or they can digest the viruses, or they can digest any kind of bacteria or viruses. Due to this way, the cell is infected. Let's suppose in this case, this cell is infected by the viruses. So they can produce a lot of viruses. Now to reduce the virus production, what actually we do? This helper T cell directly activate the cytotoxic T cell, or we can say natural killer cell. Let me write here, natural killer cell. Now what actually this natural killer cell can do? If this is bacteria, let's suppose, if this is bacteria, so then they will perform this one function. Means they will produce more helper T cell, B cell, and plasma cell, and then antibody. If this is virus, so then they directly activate the natural killer cell. This helper T cell activate the natural killer cell. Now natural killer cell will reach closer to the this infected cell, and after that they will produce granzyme. granzyme and porphyrins. Granzyme and porphyrins will produce the pores and holes to this infected cell, and finally this infected cell will kill to reduce the viral production. So that's how the production is, that's how the process are under the control. So how these processes are possible? These processes are possible by this MHC molecule. Okay, this is how the function, the whole function is proceeded by this MHC molecule. If this MHC molecule are not synthesized, so the same process will be not happen. Okay, so that's how the MHC molecule are too much important. Now one thing more. Whenever this MHC molecule is present inside the human, so we call that HLA. What does it mean? What does HLA stand for? HLA stand for human leukocyte antigen. Let me write. This is called HLA. Okay, now we call that HLA when it is present in human. Okay, now in human, chromosome number 6 have HLA genes which produce different kinds of product for HLA, means for MHC. Let's suppose we have at that position, let's suppose this is the centromere. If all of you better know the chromosomes have centromere, let me draw again for your understanding. Let's suppose this is the chromosome. Now these are the centromere, these are the sister chromatids. Okay, so let me take this one portion of the chromosome. So let's say this is the centromere, and let's say this is the sister chromatids. Okay, at chromosome, let's suppose this is the chromosome number 6 which I select. So at chromosome number 6 in human, at chromosome number 6, there are special kinds of genes which are specially responsible to produce the product of MHC molecule. Okay, so what kind of genes they have? Let me write. Now this HLA, before that let me explain that MHC have two main classes. One we called it MHC1. Let me write. They have two major classes. One we called it MHC1 or HLA1 and MHC2 or HLA2. Fine. Now this is the class 1 molecule. Okay, these are the genes of class 1 molecule. Class 1 molecule. These genes are the class 1 molecule. That's why whenever they produce the product, these products are specially for the class 1 molecule. They specially responsible to produce the product. Now each product are then responsible to process or to make the MHC1 molecule or MHC1 protein or receptor, and this is class 2 genes. Okay, these are what? These are the class 2 genes. These blue dots which I formed, these blue large dots that which I draw with a blue marker, these are actually the locus, and locus are that area which have special or which have a specific genes. So these three are the genes. Okay, I will write the name later, but first of all try to understand that. Now this is the class 2 genes. Okay, these are the class 2 genes, and these class 2 genes are specially responsible the product for the MHC class 2 receptor. Okay, now there are another class which we called it class 3, but class 3 are not too much important. Okay, and we are already discussed the class 3. Class 3 are specially responsible to produce different kinds of proteins which are specially used in the complement system and some other system. Okay, so that's why I'm not explaining the class 3 molecule here, but these two are very important. Okay, now this one genes are known as DO. This one genes are known as DP, and this one gene are known as DR. So these are the genes name. And similarly at that case, this one gene is known as A, this one called as B, and this one called as C. Okay, now these three genes DO, DP and DR, these genes are specially responsible to produce alpha and beta product. And alpha and beta product, they are specially responsible to produce the product of alpha and beta. Okay, sorry, this is, let's say this is the class 2. Okay, sorry for mistake, this is the class 1 molecule. Okay, now class 1 molecule, class 1 genes have A, B, C, and class 2 genes have DO, DP, DR. These three genes are specially responsible to produce alpha and beta product of MHC class 2 molecule, not 1. Okay, class 2 molecule. Inshallah, I will explain that what is alpha and beta chain of the MHC molecule. Inshallah, we will explain this alpha beta chain in much more detail in the next video in the MHC structure. Okay. Similarly, in this case, these three genes are specially responsible to produce the product of allelic class 1 molecules. 95 allelic class 1 molecule, 200 allelic class 1 molecule, just like that. Okay, so this allelic class, due to this way, the MHC molecule are different in different people. Okay, let's suppose in my own body there is a different MHC molecule, in other person there is a different MHC molecule. Okay, so it doesn't mean that MHC molecule have same in every person. Now it is different in every person. Okay, so what actually we studied that whenever the MHC molecule is present in chromosome number 6, then we call that human leukocyte antigen. But remember, when it is present in mice, what we call that? In mice, it is usually present in chromosome number 17. Remember it. In mice, it is present in chromosome number 17, and in mice, we call that H2 complex. So that's very important. Okay, the MHC1 molecule, this is MHC1 molecule, this is usually present in all nucleated cell, all nucleated cells, and this MHC2 molecule are present in antigen presenting cell. Antigen presenting cell. Now I already explained the antigen presenting cell means dendritic cell, macrophages and B cell. These are called antigen presenting cell. Why? Because they engulf the phagocytes, break down it into small fragment, and then they present each of the bacterial portion or each of the antigen to their receptor, and each receptor we call that MHC molecule. So each MHC molecule are known as MHC2 molecule. Remember, MHC2 molecule are always present in class 2. In MHC2 molecule are always present in antigen presenting cell, and similarly MHC1 molecule are always present in all nucleated cells. Okay, so let's suppose if this is the amino cells, they just recognize the self antigen, or let's suppose this is the helper T cell, tissue cells and some other nucleated cells have MHC1 molecules, and they just recognize the foreign antigen. Okay, so that's how the MHC molecules are performing their function. That's all about the big picture of MHC1 and MHC2 molecules. Okay, that's the function and that's everything about. But that's just the big picture and the overview of the MHC molecules. I hope you understand about that video lecture. Inshallah, in the next video lecture, we are going to explain the structure of MHC1 and MHC2 molecule in a detail, that what are actually MHC1 molecule look like and what are MHC2 molecule look like. We'll be explaining in the next video lecture, so please stay tuned and continue to watch the video. Thank you so much.